Okay, I think we'll get started. Welcome, everybody. I'd like to welcome everybody to the Fralin Biomedical Research Institute Maurice Strauss Distinguished Public Lecture Series. Welcome back for 2020. Before I introduce this evening's speaker, I want to make sure to put in a plug for the upcoming events. So next month, on February 27th, Thursday, we're having Dr. Adam Ghazali from the University of California, San Francisco speak. And he's also the founder of Neuroscape, and he'll be talking about the cognition crisis, perils and promise of technology in the brain. And then the following month, for our March, it'll be March 12th series, Dr. Teresa Woodruff from Northwestern University will be here to talk about the engineering reproduction from bench to bedside babies. And I imagine both of those will be very exciting talks and really contemporary topics of interest to everyone. So I hope you all continue to come out. I want to make sure to acknowledge and thank Maurice Strauss. I believe he's not here this evening, but to thank him for endowing and supporting this lecture series. And without further ado, I'd like to introduce tonight's speaker, Dr. Chris Austin from NIH. Dr. Austin is currently serving as the director of the National Center for Advancing Translational Sciences, or what we fondly refer to as NCATS. There are a lot of acronyms at NIH. That's only one of many. He is truly one of the nation's foremost clinicians and geneticists. By way of background, quickly did his undergraduate work at Princeton University, took his medical degree from Harvard, did a research fellowship in developmental neurogenetics at Harvard, worked, I think, with Connie Sepko for 4 or 5 years or so. And for those of you from the community who aren't in science or medicine, that's one of the great names in developmental biology and science and so forth, and he had a very productive run there. He went on and did his residency training in internal medicine and neurology at the Mass General Hospital. And then he was in medical practice in both academic and community settings, working in a variety of hospitals and primary care and urban settings, but also in rural environments, both in the United States and in other countries as well. And then also got involved in industry, working for Merck Pharmaceuticals. And there he directed programs on genome-based discovery of targets and drugs, focusing on treatments for neuropsychiatric disorders such as schizophrenia and Alzheimer's disease. His career really spans a spectrum of the very area he directs for the country, translational research. So he's one of the people that has, I think, unique and deep insights about what's important in that area. He's been recognized in many ways. One of the most prestigious, of course, is his election to the National Academy of Medicine, which he is a member. And going back a bit, he joined NIH about 2002, and he was serving then as a senior advisor to the director for translational research on the Human Genome Research Institute, NHGRI. And he was really responsible for a lot of the work that went into pulling together and implementing the research programs to derive scientific insights from the results of the Human Genome Project, which, as most of you know, is one of the most significant advances in medicine and science probably in the history of mankind. He founded and directed the NIH Chemical Genomics Center, Therapeutics for Rare Disease and Neglected Disease Program, and the Toxicology in the 21st Century Initiative. And he's been the inaugural director for the Center for Translational Therapeutics Division of Preclinical Innovation. And so before I have Dr. Austin come up, I talked a lot about his leadership roles, and I like to think of the world of science and medicine of people who play key roles in leadership and people who play key roles in doing the stuff of science and medicine. That is, scientists and physicians that are doing science and doing delivery of healthcare. And sometimes they aren't always one and the same. In Dr. Austin's case, it is really one and the same. That is, his scientific contributions have really been immense. He's involved in a number of early studies on defining genes, involved in lineage analysis in the nervous system and in the eye. Some of that work I referred to with Dr. Sepko, and I know Chris Walsh was one of your collaborators, an old friend and colleague of mine. He's been involved with cancer genetics work from the get-go. I mentioned the stuff in neuropsychiatric disease. He's done some really elegant studies looking at gamma secretase and neurite outgrowth as relates to neurodegenerative disease, looking for genes related to mental illness, such as schizophrenia. He really has been a mover and shaker in the whole field of high-throughput screening and applying this to studies to understand how receptors are involved in signaling and developing drugs to treat cancer, for example, and going on to genetic profiling and therapy. So he's a real deal. He's a whole package. He's contributed immensely in his science, in clinical care, and now he contributes immensely on the national stage as a leader. So please join me in welcoming Dr. Chris Austin.
Wow. Did I actually do all that? Yeah, well, thanks so much, Mike. It's great to be here. And one of the things that Mike didn't mention is your new dean and I went to medical school together, and he was one of my closest friends at medical school, and he and I had dinner at our house last night because he happens to be in D.C. Is one of the delightful sort of small world department things. So what I'm going to try to do is run through both some information, but also how I think about the world, the opportunities that now beset us, and actually challenge you a bit as well to maybe think a little different from how we've previous. Ooh, that's really good, but maybe I will have to sing a little bit before we keep going. So let's just dive right in here. And I really, Mike just told me that about 50% of the people in the room are not full-time science nerds, and so I'm going to try to coin a word, translate some of this into normal human language, normal English language as we go through. This is the world that we live in now. It's the world that has happened during my professional lifetime, which is that the fundamental science is really, in many ways, unrecognizable from what it was when I was in Connie's lab back in the mid to late 80s. What we can do in the lab now is just quantum levels, more sophisticated and high throughput and detailed than we could do then. But if you ask in the last 30 years, for most people, how much has healthcare changed? That is, if you think about the last time you went to a doctor now versus 30 years ago, how much has it changed? And for many fields, in my own field of neurology, it hasn't changed very much at all. If you think about the common neurological diseases, Alzheimer's disease, Huntington's disease, intractable pain, glioblastoma, et cetera, the ALS, the world is no change at all, despite the fact that we know a lot on the basic side. And so it's an interesting question to the degree that's what's on the right side, which is health. We always say advances in health are going to come from understanding of fundamental science. Or in other words, one of the purposes, one of the sort of covenants that NIH made with the public at the end of World War II was to say, well, support us, and we will understand the basis of human physiology and health and disease as a platform on which to lead to better health and intervention. And so, to the degree that science has really skyrocketed, but health is not, this gap of forming really dramatically over the last 30 years, why has that happened? It doesn't make any sense. It wasn't supposed to happen. And that, in short, is the translational problem. It's not that translation is a new thing. It isn't. People have been trying to do translation for thousands of years. It's that the opportunity on the left side is absolutely unprecedented, and our ability to actually tangibly affect health and well-being of real live human patients is really not kept pace. So how do we fix that? And I'm going to argue too that it's a scientific problem, but it's also a sociological problem and a systematic problem. As in so many things in human endeavor, as the old saying goes, what got us here won't get us there. And the system that we have is exquisitely designed to do a single investigator fundamental research like I did with Connie. It is not organized well at all to do the translation that's now needed. Okay, so here's an example of how I like to think about this. I'm a genetics guy, as you heard. So this is just a graphic illustration. This is the number of human conditions that are in a database called OMIM, Online and Mendelian Inheritance in Man. Each one of these is a disease or a human condition. And you notice when I graduated from medical school, as I was training, back on the left side off that graph, there were fewer than a dozen human diseases, the molecular basis of which we understood. And so most of the diseases that I saw in the clinic, we didn't have any idea what caused them. And just during my professional lifetime, that has utterly changed. There are now almost 6,500 diseases, the molecular basis, genetic basis of which we understand. And that is just one example, and part of this came from the Genome Project, in case you're wondering why this happened. And that has fundamentally changed our ability to think about how to treat disease. Certainly has helped with diagnosis. But if you're a patient, what you really care about is this. And the fact is that 95% of human diseases have no FDA-approved treatment at all. 95% of human diseases. That's an astounding number. You know, and I'll get back to this, but I did a calculation a few years ago. At the current rate of progress, how many years will it be before every human disease is treatable? The answer is 2,000 years. So if there's anything, and I'm going to throw a lot of numbers at you, if there's anything that tells you we cannot simply keep doing what we're doing, it's not working, that should be it. And I'll get to another couple of reasons in a second. Okay, so that's the challenge. Now you might think, well, gosh, you know, you probably know, some of you probably know, well, it takes 15 years to make a drug or an intervention, you know, once you have a target, so maybe we just need to wait a few years. Well, you would be wrong, because our efficiency of developing therapeutics has actually been decreasing quite monotonically since 1950. And let me show you this. So I hope you all know what Moore's law is. It's the number of transistors you could fit in a microprocessor that's doubled every 2 years. It's named after Gordon Moore, who's one of the founders of Intel. That is the reason why what is a supercomputer when I was in college is now in your iPhone. That's transformed every area of our lives. So at the same time, how are we doing on therapeutic development? Well, this is what it looks like. This is actually from a paper a few years ago, apocryphally named Eroom's law, which is Moore's law spelled backwards. And this is a truly terrifying graph that I find almost nobody knows about. That is, this is the number, and those of you who are economists, I actually took economics in college. It was one of my favorite courses and most useful. And you remember that economists always talk about widgets, right? They're always, widgets are how many widgets can you get per dollar? And economic productivity is defined by more widgets for the same dollar, or in other words, the price comes down for the given expenditure that you do. So what you're looking at here is how many drugs do you get per $1 billion? So if somebody hands you $1 billion, how many drugs do you get out of it? And what you're looking at here is monotonically negative productivity growth since 1950, such as that the number of drugs approved per $1 billion spent has gone down by half every 9 years since 1950. Now think about that. I don't care if you're talking about shoes or ball bearings or cars or drugs. Any industry, the efficiency of which decreases by 50% every 9 years for 7 decades is going to, so a lot of those companies are going to go out of business. They're going to merge with each other. They're going to constantly acquire each other. And the products that they do make are going to be exorbitantly expensive, which I just described the pharmaceutical industry for you. So this is not surprising that this is pure mathematics why this is happening. And it's an astounding graph, because you think about what's happened in science and medicine during this time. Right, since 1950, the invention of computers, recombinant DNA, Genome Project, gene therapy, all these things, none of it has had any effect on the decrease in productivity. Decrease in productivity. A lot of people actually have argued that all of this science has actually led to part of this decrease in productivity, because there's so much data that we don't know what to make of, that it's increased the number of wild goose chases which go nowhere, which is probably partially true. There's a lot of reasons for this. Regulatory rules have changed over this period of time and all kinds of things. But if you ask, well, when does this go to zero? And if you extrapolate out, it actually goes to zero, or you notice it's a log plot, so it can't officially go to zero, but it becomes asymptotic in about 2050. So unless something changes in the next 30 years, that has been the case for the last 70, there will be no more drugs after 2050. Or in other words, the cost of a new drug will be infinity, which is pretty much the same thing. So we can't just keep doing the same old thing, right? Because it's going to take 2,000 years, and by the time we reach 2,000 years, drugs are going to be even more expensive than college tuition, which is really saying something. So what is our answer to this? NCATS was formed in part to bend this curve. Now think about it. We're supposed to do something that invention of computers, recombinant DNA, et cetera, haven't been able to do. So actually, when I took this job 7 years ago, people just, I had a lot of things written, this guy's just, he's got a masochism complex. This is just charge of the right brigade. You know, there's no way this is going to be successful. Our thesis here, which we have data for now, is that this is not an intractable problem, but it cannot be solved by doing the same thing we're already doing and expecting a different result. Fundamentally, our hypothesis here is that the problem is the translation, for a variety of interesting reasons, has always been almost entirely empirical, that is, trial and error. We don't understand the fundamental basis of almost any part of this process. And if you think about high-throughput screening, one of the things that Mike mentioned, you know, I love robots. I love being able to we can test 3,000 different drugs at different concentrations in a week at our place, which would take a person 7 days a week, 12 hours a day for 12 years to do. We can do it in a week. Now, that's wonderful. However, think about the premise. If we have to test 3 million possible compounds to find something we're interested in, that is prima facie evidence we have no idea what we're doing. Right? I mean, it's like, I was saying, it's like hopping in your car at night and you're trying to find a 7-Eleven. You just start driving around. You have no map. So you go take 3 million different turns and you'll probably run into a 7-Eleven. But you shouldn't be proud of that. Now, the fact that we could do these more in parallel, that's great. But that is not a solution. We have to understand what the fundamental rules are to transform this process from an empirical, trial and error, phenomenological, mainly error process into a predictive science. Much like engineering is, genetics has become during my lifetime, data science now is becoming. So we're, in the immortal words of Matt Damon in The Martian, you remember this? He finds himself also in a foreign world. People are dying all over the place. What does he do? That's that critical moment where I'm going to change the words here because this is a family show. We're going to science the stuffing out of this problem, right? That's what he says. And that's essentially what we're doing. Okay, so in other words, what I was taught was wrong. So when I was at the Mass General, all these fancy places, I was taught this is what I was supposed to do. Make diagnoses as a neurologist and make diagnoses as a geneticist. Write papers and make diagnoses and everything else is going to happen by itself. Therapeutic development and implementation is a thermodynamically favored process, as it says at the end of every paper, and there will be a drug within 3 to 5 years. So silly me, I thought, okay, it just kind of happens. All I got to do is publish some papers and everything will be fine. Then I went to Merck. Oh my God. I realized that those are not the same thing. As a matter of fact, the paper you are in the foothills of the Matterhorn, right? The level of difficulty goes up and up and up and up. And furthermore, when I worked in Alaska and in Africa, I learned that just because there are drugs, if you don't have a delivery system, if you don't have a system to have people actually get these things, then they may as well not exist. And the current numbers, these two inequalities, are the translational problem. The current state of the art is that first inequality, 15 years, $2 to $10 billion, 99% failure rate. The second one, we have no idea how much it costs because nobody's ever tried to quantify it, but 17 years is the best number between something that's shown to be useful and gets to all the patients that need it. That's unbelievable. For every frickin' target and intervention, that's over 30 years. So again, we can't just keep doing what we're doing. Okay, so what the heck is translation? Translation is what we do, and we have, it's a terrible word, right? Because we're stuck with it. People use it in all kinds of different ways. So we actually had to define what we do when I became director. Fundamentally, it's the process of turning an observation, which somebody makes for the first time, and that observation may be a scientist in the lab, could be a clinician, a doctor in the clinic, could be a public health worker, in the community. Think about Ebola or coronavirus, if you think about it now, where a light bulb goes on, and the person says, nobody's ever made this observation before. If I can reverse this thing in front of me, this phenomenon in the lab, or this clinical thing that I'm looking at, or this public health problem, if I do that, I can reverse the health problem that's in front of me. So the whole process from that ideation, I have an idea, to actually making the thing, the drug, the physical thing, the drug, the device, the behavioral intervention, whatever it is, and then showing that it actually works as you thought it would work, as you hypothesized, first in controlled settings in a clinical trial, and then, importantly, where people live, which is not in a clinical trial, it's in the community. That's where people live. So we have to translate to public health. That whole process is translation. So what is translational science? Translational science, then, is like any other science. It's a field of investigation that seeks to understand what the general principles are. With the thesis, as I said before, if we can understand those general principles, then we will be able to predict what happens. The failure rate will go down from 99% to something more manageable, and therefore costs will come down too, because cost is actually driven not by the unit cost of these. It's fairly expensive to develop new therapeutics, but what really kills you is the 99% failure rate, because you've got to multiply the unit cost by 1 over the failure rate. So if your failure rate is 99%, you've got to do whatever the unit cost is times 100. So where the action is decreasing the failure rate. And decreasing the failure rate means you've got to make it predictive. And to make it predictive, you've got to understand what the general principles are. Okay, so the first point I want to make is about our solutions is to realize that we've seen the enemy and he is us, as Pogo said. So translation is a team sport, and traditionally academic organizations, universities, have not done this well. So what is a team sport? A team sport is something which requires top performers in a variety of different positions. Think about a football team or an orchestra. They're all playing different instruments, but they have to know. They all have to read music, and they all have to understand what everybody else is doing to make a piece of music successful. So wide variety of different expertise who work together to a common goal that none of them could do by themselves. That's basic definition of a team. Now, when I was in training with Connie, this was my model of what I was supposed to do. Anybody know who this guy is? Black and white photograph, bunch of crud on a petri dish. It's got to be Alexander Fleming, right? Who's famous for this Eureka Moment, right? Where you know what he did, right? He went on vacation, didn't clean up his bench, comes back and there's junk, all the mold all over his bench, and that became penicillin. This idea that scientific discovery is all about, the Lone Ranger working in the lab at night by candlelight in this Eureka moment. I've had those moments. They are absolutely intoxicating. And they're the reason I'm probably still in science, because of this dopamine rush that I got then, very addictive. And that's fantastic for basic science, and it still works in basic science. That model works really well for basic science. It does not work for translation. So let me tell you why. So this is, I always like Meg Analogy. So this is science as golf, right? Solitary exercise. Maybe you have a caddy, as well as known as your lab head. And that works great for basic science, but that's not translation. This is translation, where you got a really strong defense. That's Mother Nature, who does not give up her secrets easily. And the only reason that guy, the running back, is going to score is because he's got a quarterback who's calling plays. And number 72 there, this guy, the offensive lineman, is going to throw his body in front of the defense to allow his teammate to score. And guess what? They're all going to get credit, right? Now, this is academia. The last author, who's the lab head, Corbin, he hands off to the first author, who's usually the postdoc. What about number 72? Number 72 is the dreaded middle author, who, even if everybody scores and they get a publication, he gets no credit, and he doesn't get tenure. So, do you think number 72 is going to do anything to allow his teammates to score? Why would he? It would be against his self-interest. He's going to endanger life and limb and get no credit. So as long as we have this way of running our academic organizations, and let's just face it, incentive structures in medicine, academic medicine often mitigate against this very teamwork. And NIH is the worst among it, right? We have this thing called a PI, principal investigator, one person. That works great in golf. It doesn't work great here. So we spend a lot of time working on these things. Okay, so for those of you who don't think about drugs every day, this is drug development in 6 easy steps. Couldn't be easier. This is what most academic folks do, and they discover basic mechanisms, which are really important, because without that, there's nothing to translate. So we absolutely need this. This is not what NCATS does. We do everything distal to that. But currently, remember, these 6 easy steps, 15 years, 99% failure rate, et cetera. The thing I want you to realize about these steps, going from a testing system to be able to screen lots of possibilities to doing medicinal chemistry or something analogous to it, testing in preclinical models and clinical models, is the cost function. So importantly, you can get a basic discovery for about the cost of a graduate student or a postdoc. So 100K a year. Great. So 100K, you get yourself target identification, as we say. Okay, so now let's say you want to develop a prototype drug that you can use in cells or maybe in animals to test the therapeutic hypothesis. So if I affect this target, it will reverse whatever the model of the disease is. That'll cost you about $1 million. So it's about a log more expensive. So what that means is to do one of these projects, you got to stop 10 postdocs. So you get 10 postdocs for one of these. If you want to go to the next step to actually get into first in human, it's going to cost you about 10 million. So then you get 100 postdocs. And if you want to do actually clinical development, a cheap clinical trial will cost you about 100 million. That's 1,000 postdocs. So this is the situation that NIH is in now, where we are often held by the Congress and others to certain employment targets, literally. And so if we want to do this, we got a real problem, because the system is built to do something else. But as we try to do more of this, we have to come to terms with this. Either we got to learn to pay for this or stop talking about it. Now, granted, we want to drive down these numbers, of course. We want to increase productivity, but realize that each one of these, we're doing about, for every success, we're doing about 100 of these, 10 of these, and multiple of these. So you got to multiply the numbers for each of them. So NCATS has programs in all of these, and I'll show you a few of them. We have a division that does this in something called CAN. In the clinical phase, again, for those of you who don't think about this every day, we have these phases: small number of people, mainly safety; larger number of people looking at dosing and safety, a little bit of effectiveness; and then the definitive trial, large numbers of people, single dose or maybe a couple doses. And that leads to FDA approval if that's needed. But two things that many people don't really think about, but they are, as Mike said, if we're going to get something to help people, people don't live in clinical trials. It's a totally artificial environment where we control all the variables except one. Nobody lives in that environment. And so in order to find out whether something actually works, we have to implement it in the real world. That's where this real world evidence term comes from. Traditionally, NIH hasn't done this very well. We just kind of said, well, we got an approval, get off my back. But it turns out a lot of stuff that works here, if it's an ideal environment, doesn't work here. And furthermore, even if you can get things into clinical implementation, get them implemented into practice, they actually don't improve public health. If you think about the object lesson, the story of the Women's Health Initiative, and postmenopausal estrogen is the best example that I know of, where postmenopausal estrogen made perfect sense from supplementing estrogen in postmenopausal women to prevent osteoporosis and other things. It made perfect sense and just flew through all of these stages. And it wasn't until researchers looked at are we actually having the effect in populations of women that we thought we were, it turns out we were killing people. So stopping here, or even here, is not acceptable. You can't stop here and declare victory. So that's very much part of what NCATS does. So we have programs across all of these, both in common disease and in rare disease, and you have joined the CTSA program just recently through your collaboration with the University of Virginia. So what is our mission? Fundamentally, how do we think about this? I just painted this really, depending on your point of view, either challenging or depressing point of view, maybe it's a little both. How are we going to address this? we have all 7,000 diseases. We got to do everything from target validation to public health for all those diseases and increase efficiency at the same time. Why are we doing this? Well, history teaches us and science teaches us that the only way to make these kinds of quantal improvements, which are necessary, we can't be talking about 10%, 20% improvements in efficiency. I hope I've convinced you that is just not going to cut it. We have to look for fold improvements, twofold, fivefold, tenfold improvements. These come from innovative ways, new ways of doing things, new ways of thinking, new paradigms, new technologies, new collaborative structures. That's really what you need. And so that's what NCATS is all about, to make this process better, faster, cheaper. That's what we're trying to do. Our sort of tagline is developing systems by which we will get more treatments to more people more quickly. That's really what we're all about. Okay, so for those nerds of you in the room, and I count myself proudly among them, these are the problems we work on. So sometimes people say, well, what do you guys do all day? You don't have a disease, you don't have Alzheimer's disease, you don't have cancer, so our disease is the system. And if those of you clinicians know that every patient, you have a problem list, hypertension, diabetes, past stroke, whatever. This is the problem list for the translational patient who is very sick. These are the problems that very efficiently kill translational projects, getting out of the lab and into an intervention development and into people to show that they're improved human health. I don't care if you're talking about Alzheimer's disease or psoriasis or osteoarthritis, they're all the same issues. And that's one of the reasons that makes this tractable. They're all the same reasons. You can imagine if cancer interventions had different reasons for failure than neurological ones, then we'd really be in a pickle, but that's not the case. They're all the same. Okay, so if we solve it for one disease, if we think about it the right way, we ought to be able to apply it immediately to many other diseases. Okay, so the first thing we realized was that, as we've been saying, most folks don't understand where, Daddy, where did drugs come from? I had no idea. When I did training in all these fancy places, I had no idea I was prescribing these things. I had no idea where they came from. So I thought there was sort of deus ex machina, they just sort of fall out of the sky, right? So we realized, and this is a real problem for our congressional representatives, I must tell you, it's what leads to a lot of the silliness. There's a lot of competition for silliness coming out of DC these days, but these folks, like most of folks, scientists and doctors, they do not understand this process of translation. So we realized that we had to develop a map. And that map would be useful both for education, but also for systems engineering, because if you don't know where you're going, you don't have a map, how are you going to get there? And this was not done only by NCATS, it was done by the part of the National Academy of Medicine. And NASEM is the National Academy for Science, Engineering, and Medicine. There's a forum on drug development and translation. It's people in public sector, private sector, FDA, foundations, et cetera. And we got together to develop such a map, and we published it in some of the usual places a couple of years ago. But let me show you what it looks like. So this is the conventional chevron diagram that everybody shows, and I use it as well. I'll show it a little bit later. And we all use the term pipeline, right? This is the drug development pipeline. And what we realized was, people believe us that it's a pipeline. So think about, those are you normal people in the room, take out the scientists. The normal people in the room, what is a pipeline? A pipeline is something that brings water into your house, right? So a pipeline, okay, it's a conduit, and an entity, water or gas or whatever, goes in one end and comes out the other, unchanged, right? That's hopefully unchanged. And the progress is only dependent on the diameter of the pipe. Now drug development is about as different from that as it could possibly be. And yet I've been using the term pipeline my entire career. And then we say, no, drug development is really hard, it fails all the time. And people logically say, what is the matter with you? You can't make water run through a pipe? Are you really that stupid? And we realized people are actually taking the words we use seriously. So we realized this is not a pipeline, and we got to stop talking about that. And what does the map look like? It doesn't look like this. It actually looks like this. Exactly. And this is a low mag view. We actually have transformed this into this, which is an online version like Google has, there's 10,000 nodes in this process. And my basic science colleagues are up here in the upper left corner. It's like the board game from hell. And most people never complete it, because there's recursive loops in here, whirlpools, various things. But without this, you have no idea what you're getting yourself into. And then something fails and you have no idea why, because it seems so simple. It was a pipeline. I just had to go from this to this to this, right? So this has been really helpful for us. Secondly, we had to define, for this a field of translational science. This is a new field, never existed before 8 years ago. It's like data science. What is data science? Or genome science? They're all new sciences. So we had to define what are the defining characteristics of a translational science, and they are different from how at least I was trained. Certainly you have to be a rigorous researcher. I would argue even more rigorous than when you are simply making observations. And I want to make it clear. I think observations are really important, but they are neither necessarily reproducible, as we've found, or rigorous enough on which to build a translational program. So if you're going to do translation, you really got to have results which work every day, no matter who's doing it. No magic hands graduate student allowed here. So rigorous researcher, you have to be a domain expert. If you're going to play on a team, you got to be the best darn person on that team, because your other team members are going to rely on you. And if you mess up, that's really bad, because you're letting down your team members. So you got to be a domain expert, but you also got to be a communicator. You got to be able to communicate with your other team members. I don't know about you, but scientists, most of us, are mildly autistic. We are not good. We're trained to communicate with our cell lines. I don't know if Mike, have you ever did that? I used to talk to my cells and sing to them. I actually did. People, not so much. And luckily, my medical training helped me, and my music training actually is what did it. Where I had to develop some rudimentary social skills, but most scientists don't have that. They don't have. You have to be a process innovator, because again, we can't just keep doing what we're already doing. You have to be a team player. You have to be willing to cross boundaries, basic to clinical research, for instance, and you have to think in terms of systems for the reasons I just showed you. And we made a video. It's mainly for high school and college students, to explain what this field is all about and why they ought to get into it. We're also training people. This may be of interest to those of you here at VTC. Something called the I-Core program, which was started by the NSF. It's the Innovation Core program. What it does is take people who are trained in fundamental science and teach them how to think like an entrepreneur, how to think about whether your interesting discovery actually has a use that somebody would want to buy. It's a very different skill, starting with a problem and working backwards. Scientists are trained to at least life science, not engineers, but life science people are trained to just focus on novelty, and then we'll worry about utility later, or not at all. So this is different kind of training, but it's really useful. And so we do a lot of this through the SBIR STTR program and through the CTSA program. We also realized when we started this that because this has never been an academic discipline before, there's no scientific literature. If you're going to get into this field, what do you read? Where are the journal articles to tell you what to do? Where are the protocols? So we realized what we needed, and again, for you science nerds, for those of a certain age will remember this. This is how I learned, this is Maniatis. That's how I learned to do molecular cloning. And then this came along, current protocols, where detailed protocols of how to do experiments in the lab. And so we realized this is what we needed for preclinical development. We had some colleagues through a minor miracle at Lilly, Eli Lilly, the drug company, who said, there's a lot of bad science out there in the academic world, and it doesn't serve anybody. So why don't we take our internal tribal knowledge manual, which every company has. I had one at Merck, how we do things, and we're going to take out all the Lilly references and put it on your website. Unbelievable. I mean, because most companies treated this as proprietary. That was about 15 years ago, and now there's an international editorial board that's freely accessible as an ebook on the NLM website. I meant to put the URL in there, but it's in the next slide. And the important thing is it's not only rationale, but detailed protocols. If you get into this for the first time and you want to do something, this is how you do it. And these are the statistics, these are the methods, these are the protocols. And so it's now 46 chapters, almost 1,400 pages, and it's dynamically updated all the time, like current protocols is. The big difference is you don't have to pay for it. It's all free. We also, for those of you who are oral learners, we have a bunch of workshops that are available online. A lot of the same sort of topics, but they're in lecture format. Okay, so on the science side, it's going to get a little heavy here on the science. So for you normal people, again, bear with me. The scientists will probably go into some sort of state of nirvana at some point. So, what is the problem in the preclinical space? The problem, as we described it, is not that there are not fantastic, smart, committed, fundamental scientists out there. There's many of them in this room. The problem is that they have neither the training, nor the knowledge, nor the facilities, nor the funding, nor the incentives to be able to take those discoveries and move them down this non-existent pipeline. And I just said this is the chevron. I told you it doesn't exist, but for simplicity purposes, I put it up here again. And so we created a very different organization. This is the intramural part of NCATS, which means it's basically an academic style group at most parts of NIH. They're essentially professors and they have tenure and all the usual stuff. So this part of NCATS has no tenure, has no tenure track, has no principal investigators. Everybody works in project teams, and every project is a collaboration with somebody somewhere in the world. Now, when I go to universities and I say we have no tenure, usually somebody has an MI and has to be carried out. And it's not that we don't like tenure. The problem is that the incentives that tenure enforces are antithetical to the skills and behaviors that are necessary for success in translation. So that's why we don't do it. But our folks are world experts in translation, but they do not have disease expertise in all 7,000 diseases or every target. So the mistake that companies often made, and Merv makes this too, is to act like they were. And so we said, no, we're going to let the academics be academics, the folks who really understand their target in the disease, let them do that, but put them on a joint project team with people at NCATS who really know the translational part. Joint project team, they have to put a project plan together, go-no-go decisions, milestones, and as long as they keep meeting their milestones, they get funding until the point that the goals are achieved. Okay, so there's multiple entry points. Those are people who were stuck at various points up there, and they go through peer review, and they go into one or another programs, and there are programs across the spectrum. And then what comes out at the bottom are deliverables, and those deliverables are useful to the individual academic PI to move their project down the translational spectrum, but also from our point of view, they're dual-use projects. They tell us something designed to tell us something general about the process, so that we can learn the general principles to make this work better. So let me just give you one example. I know that you're interested in oncology and pediatric neurology, so I chose this. But there's 179 projects going on currently in this organization with every area of biology and disease with people all over the world. So this is just one example, but it really is illustrative of both the science and the sociology. So this happens to be a really, really awful pediatric cancer called DIPG, diffuse intrinsic pontine glioma. It's an invasive tumor of the brain stem, which means the base of the brain down here. It's where all the really essential functions of breathing and blood pressure and heart rate, autonomic functions, all that stuff live. And all of the stuff that comes down here to communicate to the body has to go through the brainstem. So you can't operate on this space. It's just you can't. There's no real estate here, which is expendable. So the only treatment is radiation. There's about 450 kids diagnosed a year. You see the median age is 6. Median survival is less than a year. 90% of these kids die within 2 years. Only 1% live more than 5 years. It is an absolutely horrific disease with no treatment. So, why is this a success story? Because I wouldn't be telling you if it wasn't. It worked because we did this in almost every way different from how we would normally work. Normally what it would be is one lonely PI working again by candlelight on the Alexander Fleming model, and that poor person would have to learn all of these other fields, which is, it just doesn't work. It's impossible. It's like having 11 quarterbacks on the field and expect you're going to win the Super Bowl. It's just not going to happen. So in this case, there were 3 oncologists from molecular to clinical. One at Stanford, one at, she was at NCI, but now she's at the Farber and Boston Children's, and this guy at Hopkins. And they all had different expertise. And then within NCATS, there are, say, 1, 2, 3, 4, 5 different expertise, informatics, chemistry, automation, compound management, biology. And this is what we find in every team. In the early stages, it's around 10 different disciplines. The whole kit and caboodle, it's about 20 different disciplines. 20 different disciplines that all speak 20 different languages. Requires a lot of project management, like the football team, you gotta have a coach. That again is antithetical to the way most science is done. What did we do? And this is a little bit of nerdology here. We have a really unprecedentedly large collections of all the approved drugs ever approved for human use worldwide, but also a lot of investigational drugs. So there were a little more than 2,700 compounds that were tested against 6 different cell lines developed from autopsies of children who died of this disorder. And what you're looking at is multiple concentrations. So you got to multiply this by 10 different concentrations times 6 lines. You could never do that manually with the robotics we have. It's actually relatively straightforward. A couple of compounds, one of which had been approved, another one that hadn't, came out of this. We then looked for which ones might actually get into the brain and which ones were quite potent, because you want both of them potent and get into the brain. That required some different people. There were 22 compounds that were, but then we realized that, like in most infectious disease, in disorders where the disease is constantly mutating, you got to have combinations of drugs to work. So then how do you test combinations? We have developed a technology internally to be able to do this. So we tested almost 10,000 different drug combinations to find combinations of drugs that work better together than separately. And that required a different set of expertise. This is another NCATS person. And then we went from those combinations very rapidly to another team member, brought him in because we needed his expertise. Another guy at Stanford does animal models, looking at these drug combinations in animal models and showing that they actually do have a rather dramatic effect, extending survival through implanting these cell lines into animals and then treated with these combinations. And the combinations extended animal survival just as we hoped. And then these led very rapidly into 2 clinical trials that are now ongoing. One is a phase 1 trial of this one drug, and then there's another new formulation. This drug hasn't been approved, so the IND submission is being done. And the clinical trial with the combination of them is currently being designed by the Farber and Stanford and Oregon Health. So all this happened remarkably fast. And so what happened, of course, there was a lot of appropriate celebration, and my favorite headline is this one. It really is true. It was a near hopeless childhood cancer that succumbed not only to this drug duo, but it succumbed to a model, and what I like to call is the NCATS model of doing something. It's something that we're all well aware of. It's called teamwork, and bringing different disciplines together. What we constantly find is what's impossible for one person is trivial to another, and vice versa. And if you bring those very diverse teams together, you can make headway incredibly quickly to do things that everybody thinks are impossible. A couple of other examples. We're very focused on what drugs fail in development. A number that you probably want to keep in your head is that we're all excited when a drug goes into people for the first time. That's all wonderful. But the fact is that 9 out of 10 drugs that go into people are never approved. 9 out of 10 times, even after all the work, often 10 years of work, $100 million that goes into this, you go into people, 9 times out of 10, it fails. Why? That's what this graph on the left shows. 55% of the time it's because there isn't effectiveness in people that was seen in the animal model, and about a third of the time it's because there's toxicity and adverse effect in a human that was not seen in an animal model. General lesson, rats are not humans. Absolutely robust and reproducible finding that we are very fond of. You know, and we kind of joke about it, but when you think about it, you know, those of you who are scientists, we actually kind of implicitly think they are the same. You know, they're model organisms. Models for what? Models for humans, right? And they might be little small things, but, you know, a mouse is just a small, furry human. Wrong. You know, in fundamental science, there is a lot to that, but not when doing therapeutic development. So then we thought, well, gosh, you know, shouldn't there be a better way of, with all the technologies that are out there now, a better way of using human cells that would be more predictive. And so we and a lot of other people have done not only these 2D cell things, we do a lot of those still, but these things called spheroids and organoids, which are self-organizing groups of different types of cells, which sometimes look like a real tissue. We do a lot of 3D printing. This is actually 3D printed skin that we made in the lab. And those of you who have ever seen, you know, Wikipedia picture of skin looks just like this, but we actually made this. And then what I want to just talk about for a second, which are these organs on a chip. The idea here is these are all useful, but for different things. These are very high throughput, but not terribly physiologically complex or necessarily predictive. And then organs on a chip are relatively low throughput, about the same throughput as a rat currently, but they're made out of human cells. And so might be more predictive. Okay, so what is a tissue chip or an organ on a chip? Sometimes it's called. It is not pate. You know, when we say liver on a chip, we're not talking about pate on a potato chip. We're talking about, that's what we're talking about. And what these are engineered systems, usually on a form of plastic, where you etch into the system, into that plastic, channels which mimic a human organ, the function, the structure of a human organ, and then you print tissue, you print cells, human cells, either derived from stem cells or primary cells, into them. And again, for the nerds in the crowd, these are all the things that these systems have. These are all things that the organs in your body have now. That you're sitting here, but for most of my career, I've done a lot of cell work, and basically you have none of these other things. And it's not that was bad; it was all we could do. So I don't want to diss the work that I and many other people did. But then we asked, well, gosh, you know, if isn't it possible with the convergence of stem cell technology and 3D printing and microfluidics and imaging and cell sensors and computation, that we could create little mini human organs that represent the structure and function of all human organs and use those to predict safety and effectiveness of novel drugs, not a rat. And Teresa Woodruff, who you're going to have in, make sure you come to this thing. She is incredible. She is one of our chip people. She has created, and I'll give you the punchline here so entice you to come. She has developed a tissue chip platform of a functional human female reproductive system that cycles on its own on a 28-day cycle, and you put in mouse oocytes, because we can't use human oocytes for obvious reasons. You put in the mouse egg oocytes, and they're affected by the human cells from the fallopian tube, and the cervix, and the vagina, and the uterus, and they cycle on a human cycle, 28-day cycle, even though they're mouse eggs. It's unfreaking believable. And I got to tell you, as a developmental biologist, Connie taught me this would never work, but it does, for some really interesting reasons we could talk about if you're interested. But the bottom line is, this went from nothing to a gigantic field in 8 years. Why? Because of the same fundamental principles of teamwork, management, multiple different technologies coming together and people coming together, involvement of FDA from the very beginning. Pharma was involved in this in the very beginning because they're going to be a lot of the users. these things have different shapes and sizes. Some of them are like these. This is a lung because a lot of these things. These are blood vessels. This is actually a gut, which is innervated with nerves actually, and it actually peristalsis by itself. It starts to. It's really kind of nausea-inducing to watch the video. And Teresa has one of these. But the important thing is just to give you another sort of tickler here. You notice that this lung is expanding and contracting, and that's one of the characteristics of human tissues, right? They have physical chemical properties, or mechanical properties. Your lungs are constantly expanding and contracting. Your heart as well. Tissues have blood flow through them, and it turns out that one of the secrets here to having these things be able to create human organs, which are actually mimics of what you see in the body, is you have to have that mechanical perturbation happen. So when the people who made the lung chip, when they just made this wonderful, bilayer, made it look like capillaries and alveolar cells and all the things in your lung, the cells didn't look like normal lung, and they didn't behave like normal lung. When they started stretching to mimic breathing, the cells differentiated into terminal differentiation. They started behaving like a lung normally would. The liver, which is on the bottom, that's that red and green one there. Unless you can make a sinusoid, which is the fundamental unit of productivity of the liver, but unless you have blood flow, they don't respond, because liver cells are like blood. And it can't just be continuous flow, because remember, the heart goes through systole, diastole, right? So blood flow is pulsatile. So you got to do pulsatile flow. When you do that, they differentiate. It's really a little creepy, I must tell you, but because if you're a cell and there's static, the blood isn't moving, what does that mean? It means your person's dead. So why who cares? I don't have to be differentiated anymore. And what's amazing to me about this is that this even happens when the cells are derived from induced pluripotent stem cells. So we've taken a skin cell, erased its memory banks, so it doesn't remember anything about what it was, and you differentiate them again into a different cell type, and it still acts like this. It somehow knows what its neighbors are doing and is able to actually differentiate. It's quite extraordinary. And it illustrates one of the things that we find in translational research all the time, is that by doing this kind of work, we discover fundamental principles we never would have known in any other way. So we discover a lot of basic principles, but we do it by working backwards. Okay, so we're now doing this for disease model discovery. The way what I used to do, and I still do to some degree, is if you have a disease gene, what do? Well, first thing you do is you knock it out in a mouse, and then you put the human gene into the mouse, and you convince yourself that a mouse with 19,999 mouse genes and one human gene is a good model for that human disease. Now, it's the best we could do. And it's discovered a lot of really great things. But wouldn't it be better if we had these tissue chip platforms to model human diseases? And so we're doing that and discovering things that we never knew before. And we're putting these together. This just happens to be an example of a person from Colombia, where what I'm showing you here is these are 4 different organs. This is a skin, a heart, bone, and liver. And these are all in one platform. And they have vasculature underneath them, much like a capillary bed normally would. And you can look at interactions of all these different organs and metabolism of drugs by the liver, and then cardiotoxicity or effects in skin or the bone. And so what this becomes is question that we're now addressing: is this a new connotation of the word first in human? So making individual tissue chips of individual people, because remember, these are made of stem cells, so you can make a chip of all the people in this room and look not only at human response, but you're looking at individual human response to safety and efficacy. Never been able to do that before. And as we get to these therapeutic modalities that are increasingly personalized, like all these things we're talking about, this is really important. So we've started a program now called Clinical Trials on a Chip. And the idea here is, could we take these chips into clinical trials and provide information on individual or subgroup responses before the treatments or in parallel to the treatments. So have a clinical trial and a chip trial going together, you get the same results. To help with streamlining, addressing the safety and efficacy issues I talked about during drug development, streamlining this process by selecting people for clinical trials who are most likely to respond and least likely to have an adverse effect, and addressing this critical need for small population trials. I think the last thing I'm going to tell you about is this one. So one of the problems that we have in translation is a literal translation problem. That is, if you think about the language, the idiosyncratic language that each of these 20 groups that I told you about use, if you think about the language that geneticists use, so I'm a fly geneticist by training, and fly geneticists like cutesy little names for their genes, and so early on I worked on two genes named rutabaga and dunce, because it was a learning mutant, so it was dunce. My personal favorite is one called cheap date, which is a fly that gets drunk on a small amount of alcohol. But that doesn't tell you very much about what the gene does, right? It's a cutesy little name. But you think about physicians, they're just as bad, right? What does Alzheimer's disease tell us? It doesn't tell us anything except that Alzheimer described this patient. Parkinson's disease, Huntington's disease, cancer. Cancer means crab. Last time I checked, there are not crabs in any cancer in a human. So the names are just inexplicable to either to themselves, they don't mean anything to themselves, and they're certainly inexplicable to any other community. Think about pathologists. I love pathologists, but they have things called, they have lesions that they refer to as being currant jelly-like, and caseating, like cheese. These names that have sort of grown up over the years. Now just imagine you are an intrepid researcher, and you want to go from a fundamental gene to a process in a cell, into a cell type, into a pathway, into a cellular process, into a drug, and the drugs have all these incomprehensible names too, into a disease. And let's just say that you have a drug, which you think, well, you know what it does, but what you'd really like to know is what are all the other diseases that it might be useful in? So how do you currently do it? You put the gene, you put the drug into Google and you press go. And you hope to heck you come up with something that gives you a hypothesis. What you really would like to know is, okay, so for this drug, I want to know every cell type in the body that has this gene active in it. I want to know every pathway that gene is active in. I want to know every disease in which there is a mutation in any of the genes in that pathway that's targeted by that gene, by that drug. And I want to know all the diseases that are connected with any of those things I just mentioned. You cannot ask that question now. Those are conditional questions. Google is great at answering, is A related to B? But those of your clinicians know that's not the kind of question you ask. What you ask is, under the conditions of A and B, and sometimes C, then if we have an observation of D, how likely is F or G? That's a classic clinical question. And you want to know all the possibilities, right? So that's what Translator is trying to do, to cross what Chris Shute likes to call the chasm of semantic despair. We have the basic people over here, the clinical people over here, and this poor researcher in the middle who's just trying to connect the two. And so we have this vision a number of years ago to create an open source data translator for the research community to computationally explore knowledge, research hypothesis, provide tools that augment human reasoning, because these are dependent relationships, multiple qualified dependent relationships, which the human brain is absolutely incapable of keeping straight. And if you know anything about how doctors make diagnoses, you know what I'm talking about? The average doctor makes a diagnosis, a provisional diagnosis, within 30 seconds of listening to the patient, and becomes increasingly inured to any data to the contrary, right? I mean, this is just how human brains work. Not optimal, right? But we can't just beat on the medical students to do better. We got to give them tools to do this. Some of this you could call AI, I suppose, but it's really what it requires is a really interesting idea that, again, nobody has ever done. That is, what if you enumerated every gene, every pathway, every cell type, every cellular process, every drug, every disease name, and then you remember when your kids or you were in school, you had those things of what's related to what, because you have a list over here and a list over here, and you draw lines between them, apple, orange, car, truck. That's basically what we're doing, except you got hundreds or thousands of entries, and you want to make the many-to-many connections across 20 different columns. That becomes a 20-dimensional computational problem. 20 dimensions, like life is in 3 dimensions, then we got time, that's 4 dimensions. 20-dimensional mathematical problem, which makes the computational people, I must say, drool. So we get really good people, they love things like this. And then the idea is that you would be able to dive into that matrix, that 20-dimensional matrix in any place you want. If you have a cell type, you have a drug, you have a disease, whatever, and be able to relate any other level of how we think about living systems defined by you. So when we started this about 3 years ago, it was another one of those crazy ideas. We had no idea whether this was work, so we did a 3-year feasibility phase, and I told these folks, you got 3 years. You got to show me, is this doable or not? You got to demonstrate a prototype, and if it is, how much is it going to take? How much is it going to cost? How long to develop a full translator? And again, I probably wouldn't be telling you this if they hadn't exceeded their milestones showing that this is feasible. And the lesson, if you ask, why did this happen? You know what the answer is by now. Seamless teamwork among members from communities that never talk to each other. All of these different communities, there's 20 different expertise, everybody from fundamental genetics to people who do environmental exposures and public health and everything in between, from all these different institutes and centers. And they all work in teams which are designated by colors so that they don't become tribal, because humans love to do that. So we have multiple color teams. They actually ran out of visible colors. So we have red. So then we have infrared. And my favorite new one, the best new one, is they have Team X-ray, because they're running out of wavelengths. And you can do things like finding unanticipated patterns in clinical, open clinical data. And I just want to show you how fast this happened. So we had the first face-to-face meeting about 3 years ago. This was an idea then. And we in 3 years have gone from an idea to these separate project teams solving multiple problems along the way, increasingly by hackathons, which are getting these folks together and just these nerds see with each other for a week, and they just code out. It's really quite. We got to invite a sociologist into this, because it's really a bit alien to most human behavior. But the important thing is that it really works, because you got multiple heads working together. And what I want to show you, this thing at the top that went by, I want you to look at this, because this is an individual problem in real time, sped up, that was solved by individual members of the team. Each of those dots is a different member of the team, and each of the colors is a different expertise. And we actually tracked, because we're interested in how does this actually work. Each of those movements is a member of the team going to another member of the team, asking whether they have information that can help. Then they form different nodes and different levels of activity. And you'll see finally the structure which solved this particular problem had this final structure, which was unique to this particular problem. So the next particular problem, you take the same number of people and you throw them out on the blackboard again, and they reassort in different ways to solve a different problem. It's really quite extraordinary. But this is so productive that we now have half the people, there's 100 people working on this project. Most of them are old hoary academics who tend to want to get paid for what they do. Half of them we don't even pay. We don't even pay them, because they say this is the best project they've ever done in their whole career. And they want to do it for free. It's just extraordinary. So we had this article in Stat last year. It was an insult, though, because they said, we're trying to build a Google. We are not trying to build Google. If Google can answer the question, then we're not interested. That's actually that is the criterion that we start with. If Google can answer the question, it's too easy. So what we're trying to do is but this is what's really been great on the sociological side. The collaborative nature of the products, basically having a menu of great ideas from all the smartest people around the country. This is why scientists go into science. And our job was to create the ecosystem, the environment in which this can happen. And so the point I keep coming back to, the problem that I started with are really big. And I'm going to give you one last one I think about. But they are solvable. Okay, so really quickly, last one. Okay, scope of rare diseases. I told you there's 7,000 diseases. 80% of them are genetic, most of them are in childhood. There's 250 new ones being discovered every year. It's quite extraordinary. Individually rare by definition, but cumulative they're common. This is a public health problem. 8% of the population has a rare disease, same number as type 2 diabetes. I dare say most of you probably have never thought about this before. Very high costs in direct patient care and loss of productivity. The average diagnosis takes about 10 years. These children and their families wander in the wilderness until they happen to run into a clinician who saw one of this case in medical school, says, I know what you had. By that time, the patient's going downhill, or sometimes has even died. As I said, only 5% of them have any treatment. So what's the solution to this? Stop treating these like they're individual diseases. They're not. They're all connected with each other. And if you address the problem in this way, we're quite confident we'll have more progress. So platforms. We have a program called the Rare Disease Clinical Research Network, which those of you at VT should think about. These are teams of folks who work on groups of diseases held together by commonalities biologically, and some of these you'll probably recognize. I just want to show you one of the reasons we're doing this. So collaborative development of therapeutics through these platforms, that is, technologies are going to be applied to many different diseases, and I want to show you what's at stake here. This happens to be a project that we did, it's completed now with a company called Agilus, and got bought by PTC, but this is a child with something called AADC deficiency. It's a typical pediatric rare disease. This is a 4-year-old who is barely responding to her mom. Doesn't reach out, grimaces, but not clear how much of this is voluntary. This child will go downhill from here. Most of these kids virtually all of them are dead by their 10th birthday. No treatment. This is after the one treatment with a gene therapy. A year later, this is the child, and a year after that, this is her in kindergarten, and you would be hard-pressed if you watch this to distinguish this child from all the other kids in kindergarten. There is no reason this is a special case. That's what's at stake. Okay, so we created a program to do this, platform gene therapy program, which aims to address this bottleneck, doing one disease at a time, developing, again, for you nerds in the crowd, adeno-associated virus vectors that are generic, and doing all of this in an open source way so that multiple diseases can be treated in the same way. And lastly, I wanted to just say something about the CTSA program. You're now part of this. What is this? It's the NIH's biggest single program. It's about $550 million a year, so welcome to the group. It's run by a guy named Mike Carilla, and it collaboratively facilitates translational projects. The lower half of that problem list that I showed you a little while ago. We do a lot of training, scientific and operational innovation to improve the efficiency of the clinical parts of the translational process, and importantly, fosters the creation of academic departments of translational science, which have different promotion and tenure structures, different incentive structures, and are built around teams. Okay, so, sorry. One of the ways that we've done this is thinking about this, again, in a systems way, using that map to say, what are the rate-limiting steps to this process being successful? It's a multi-step process. Rate-limiting steps, one of them early on was single IRBs. It could take 2 years for a clinical trial to get through the 20 different IRBs that it needed to. People become ineligible and die during that time because we can't get lawyers to agree to a protocol. Really? And that was said to be an intractable problem. Don't even try. We said, sorry, we think it's solvable. And here we are, it's solved. by us working with the leaders of the translational CTSA program. There's now 700 institutions that have signed this treaty, and they can all rely on each other. We then attacked the recruitment problem. Most clinical trials take 5 to 10 years to recruit. About half of NIH-funded trials will do that for 5 to 10 years and then give up. Give up. We never have any data, so that money is wasted. Young people's careers are wasted. Why? Because they can't find the patients to be in the clinical trial. There's sick patients everywhere. So what's the problem here? It was nobody's job to solve it. So we took it on, and we're not totally solved yet, but we're pretty close. I gave this talk at Harvard a little while ago at the MGH. So you guys are on here, though. Here's University of Virginia in the bringing to act. So you're on the launch pad. You should make sure that you guys, given what you told me about the clinical network, just make sure that you guys are included in this, because it's really important. But you notice there's 125 million people taken care of already, and we can look across all these 125 million people and find people for clinical trials in an instant now. And then we created a trial innovation network to develop and demonstrate innovations, scientific and operational innovations, which increase the efficiency and effectiveness of clinical trials, get them developed on time and on budget, which almost none of them do now, novel clinical trial designs, IRBs, recruitment, all of that kind of stuff. And I keep saying the last thing, but this really is the last thing. The HEAL program. So this is, and I throw this in because I know you're interested in addiction. This is the NIH helping end addiction long-term HEAL program. We're involved in 3 of these, preclinical and clinical pain and expanding therapeutic options for addiction. I want you to know about this because there may be people in the crowd who want to apply for this, and it's open right now. This is an effectiveness research network, not efficacy, it's effectiveness of existing therapies or novel approaches. These could be behavioral or pharmacological or devices to manage pain while reducing the addiction risk or opioid use. And these are meant to be very practical for practitioners in the community who have been told, don't use opioids anymore, but you got Mrs. Jones in front of you who's got chronic pain. What are you supposed to do? And there's very little guidance out there for practitioners. And so this is being run by the CTSA program through the Trial Innovation Network. And so we're soliciting now 2 additional trials. We're focusing on chronic pain in primary care settings and acute pain delivered in emergency departments, hospital settings, and where there's best practices is lacking. This is all of the sort of guts, but the important thing is that what comes out of this should be of use, immediate use for people in primary care, emergency departments, dental clinics, hospitals. For those of you who think you might be eligible, the deadline is March 24th, so please write this down and send us an application. And that really is the end. So what I've told you is, I started out with some pretty depressing stuff. But what I want you to take away from this is that it really is possible to bend the E-Room's law curve. And it is imperative, I think, from a scientific perspective, from a medical perspective, from a taxpayer return perspective, and yes, from a moral perspective, that given the fact that we have this opportunity now, we know we can do this better. We have to together change the system to make this the norm in more of the kind of work that we do. And I'm delighted to hear what I've heard so far just in the afternoon, that you all are with us in that mission. And I look forward to working with you more now that I know you're here. So thank you.
After that spectacular talk, I know we're running a bit late, but allow for maybe just 1 or 2 questions. So, questions? Yeah, Anthony.
So, I think one of the things that comes back all the way through all the examples you used is the issue of scale.
Yep.
And, which is a popular buzzword now.
Yep.
I think what it really means is that there's an enormous amount of information density for each issue. And it seems like the thing that has to be done at both at the expert level is basically figuring out ways of combining that expertise to get a singular answer.
Yep.
And, you know, I mean, everybody's ever had to find one little conclusion in the literature knows
Yep.
how long that can take.
Yep. Yep.
So how do you see scaling this up? Because clearly
Yeah.
you need human expertise, but you also need computational
Yeah.
skills, and you also need a sort of a change of how people look at problems and reliability of data,
Yep.
et cetera.
Yep. Yeah. Well, it's the question of the hour now that we have done, as I like to say, the first 2 of 3Ds. Everything we try to do, we try to develop new ways of doing something, demonstrate that it's useful, and disseminate it so that other people can use it and it can be applied at scale. And each one of those is really hard. We've tended to not really think about dissemination. We sort of put it in a journal and somebody else's problem, but it is our problem. Some of it is a cultural issue and operational issues. We've been talking about it. Some of it is a data issue, and it's really what translators trying to get at. But some of it is also it's an efficiency of it's defining how these teams actually can be more productive in a way that can be exported to somebody else. I'll give you an example. So just this year, the therapeutic development part, the part that goes lead to IND, first in human, that program has a budget of about $30 million a year, had 9 INDs in a single year, which is absolutely unheard of. I mean, most companies, if you have a $100 million budget, you get 1 IND in a year. So for 9 INDs in a single, IND is an investigational new drug application, you're going into people for the first time for an untreatable disease. So then the question that we're asking is, okay, that's a phenotypic observation. What's the mechanism? Why does this happen? And so that's actually what we're looking at, because if we're going to then disseminate this to others, we have to know what is the cookbook to allow them to do that. But NCATS it's an interesting organization because we're fundamentally, to your point, we're an integrative center. Whereas most of NIH, all the rest of NIH really, is a stovepipe sort of function. We have, absurdly, we have a bone institute and a muscle institute and a liver institute. In 2020, you love in our field, right? We have a mind institute and a brain institute, and they're separate. I mean, come on! And this actively impedes science. So, so everything that we do is integrative. So we work this way, not this way. And I would say the biggest problem we have is actually in all the rest of my colleagues, who I love dearly, but they are responding to the same kind of PI, quote unquote, incentives as PIs in an individual organization. The directors of institutes get credit for their institute's effort, not for teamwork. So our biggest challenge is getting our colleagues, even when we show these ridiculous levels of productivity, to work with us.
Thank you so much for your presentation. As we talked about, so many diseases result from people's behavior.
Yep.
Have you contemplated building your translator out so that there's behavior on the other end?
You could be really helpful with that. Welcome to the team. And as I said, we're not paying you. We really are, in that. It's a really interesting question, so thank you for asking, because when you think about the intervention part of translator, it's drugs. It's not even devices, which is absurd, as we were talking about earlier. So, thank you. You're welcome to the team. You may regret saying that.
Well, first of all, I'd like to thank you for one of the most intellectually brilliant and comprehensive talks I've ever heard in my life.
Thank you. Thank you.
And there are so many things I'd love to pursue, but I want to ask an implication question. And this is really going to the idea of principles that can be extrapolated to really complex systems.
Yep.
I'm a scientist, I'm a university professor,
Yep.
I love universities, but I also realize that we are somewhat like dinosaurs, particularly when we talk about the kinds of complexity
Yep.
that you are alluding to.
Yep.
Given your success in pulling together institutions and teams and disciplines, do you have a couple of insights that you might share about the implications of what you've done for the way that universities go about preparing people to become productive engagers
Yep.
in this new level of science?
Yep. Yep. Yeah, it's a great question. And thank you for the question. It's something that we think about a lot. Yes, we have training programs that teach people to have all those characteristics that I mentioned. So, if you're, sometimes when you start talking about team science, people immediately say dilettante. Now, if you've watched football over the last few weeks, you know that each one of those players is incredibly good at their position. And so the first thing you got to do is have it be clear that the values that we have in universities need to remain for individual excellence. It's that we need to add on to that the ability to understand at least one other discipline. As we often say, a mantra is you have to be a jack of all trades to some degree and master of at least two, so that you understand what it's like to go from, if you ever look at a biologist and a chemist talk to each other, it's like husbands and wives talking to each other a lot of the time. They speak totally different languages, but they can, with some practice and counseling and time, they can actually learn to communicate and do productive things together. But you multiply that times 20, and so there are skills that we have to teach people about how to do that. And I think my training in music actually helped me with this, because if you think about how a musical group works, everybody has their own function, but they all know how to read music, they all have theory, they all understand fundamentals of technique and style. And so that is, and only when you have that can you have, depending on, you can have a string quartet or an orchestra, but that's very different from training people to be a soloist only. And so those are skills that we teach people, and they are teachable. But what I'm worried about is that we, NCATS, for a variety of really important but so far intractable political reasons, we actually can train people, but we have no way to support them in their career. We're the only institute that we have a very robust training program, about 900 trainees, but we have no RPG program, no R01 program, no way to support clinical and support investigators in their career. So within our bubble, all of these skills and knowledge and attitudes really work well, but then they have to go to a non-permissive environment of the other institutes, so they have to de-differentiate and go to all these behaviors that we told them are not productive for what they want to do, but if they want to get a job and get tenure and pay their mortgage, they got to do it. So that's the problem, and universities, I think, are really agnostic. I think that is, if there was a way for NCATS, just as an example, to support departments of translational science.
There you go. That's what I was going to say. Why don't you guys put the money out there to us?
Then you move the cheese and the mice move. And so,
It's behavior.
yes, behavior. It's incentives. But the other thing, which I think is really, really important, which I think about a lot, which the universities do have to unlearn, is that starting at the end of World War II, and I'm a poster child for this, we convinced ourselves that reductionism is the answer to everything. And so when you think about what gets the coin to the realm in academia, universities now, it's fundamental science that goes into Cell magazine, right? And that's wonderful. but they're simple systems. They're working in yeast or single cells, whatever. Whereas the people who work in humans, outbred humans, which is orders of magnitude more complex and more difficult, those people publish in third-tier journals and maybe get tenure in a clinical department, but they are way down on the university intellectual pecking order. And I often think this is like, think of it, if we organize the Olympics like this, just imagine how this would work. You know how the Olympics, you have a multiplier for degree of difficulty? So if you do, if you're a diver and you do, really great cannonball off the low dive, it's great, but, you're gonna get a score, but you get no multiplier. If you do a back twist with a flourish off a 30-foot diving board, high degree of difficulty, you get a multiplier. In academia, it's the other way around. You actually get a multiplier for doing the simplest things. And when you go to the most difficult things, you actually get subtracted. Your credit gets divided. It's totally backwards. And I can understand why we did this, because we said, well, okay, in order to understand, Humpty Dumpty, we got to take Humpty Dumpty apart. Fine. And then we'll put Humpty Dumpty back together again. But then we forgot, well, Humpty Dumpty will take care of himself. And we have a whole bunch of people who are interested in parts. And we have whole departments that do parts. And they think all their parts are more important than the other guy's parts. And so we have to reverse that cultural pecking order as well as the operational way that we incentivize people and pay people, so that we're incentivizing them to put Humpty Dumpty back together again, if we're interested in the health of Humpty Dumpty. Which,
so this is a wonderful discussion. We can go on much longer, but due to the time, please join me in thanking.
yeah. That's great. Yeah, thanks so much.